Good evening to all. How are you? Today, now in this session, we will try to know about urine concentration, urine concentration mechanism, urine concentration mechanism. Previously already we studied about that structure of kidneys and the functional unit of kidney. That those are the nephrons and urine formation, and also about the filtration process. So related that now we should know about urine concentration mechanism. Urine concentration mechanism. Okay, so what is the main function of kidneys? Kidneys main function is they have to maintain proper osmolarity in our body. So that is regulation of body fluid. So related to that, we have to discuss two situations here. That is the diluted urine and concentrated urine. Diluted diluted urine and concentrated urine. Diluted urine means whenever more availability of water, whenever we intake more amount of water, then at that situation we will pass more volume of urine through the urine. We will send out more volume of water. At the same time, opposite to that, when we deprive to water, lack of water conditions, and whenever at the time of busy we will not take sufficient water, then the kidneys filters again to that body fluid and return to our body. The in that situation, we will pass, uh, uh, we will mixture out less amount of water, less volume of water, whatever the diluted urine or concentrated urine. In those situations, the waste materials are same, only slight difference in the that situations. Though that is the 600 milliosmol per liter a day. So these are the waste substances, waste materials which we pass out through the urine per a day. In plenty of water available, we will mixture more volume of water. Then when less amount of water is available, when deprived to water, there is no sufficient water we will take. In that situation, we will mixture concentrated urine. So in both situations, the waste materials in that urine is the same, only slightly difference we can see. So before related to that, uh, the concentration urine concentration mechanism. So inside the kidney already we know that there will be nearly 1 to 1.2 millions of nephrons are present. The main function of those nephrons is to filter the blood and also to maintain the concentration gradient. How this concentration gradient creates to to mixture to pass out concentrated urine, that kidneys inside the kidney, those nephrons have to maintain the concentrated gradient in the cortex region and medullary region and inner medullary region and papillary region. All these all this concentration will by the nephrons. How this concentration creates, how it maintains, we will see in that um, now. So this is the our this is the structure of nephron. Structure of nephron. This is a Bowman's capsule. Bowman's capsule. This one is the PCT descending loop of Henle. This is the hairpin structure. This is the ascending loop. This one is a DCT, and this is the collecting duct. Collecting duct. So to inside the kidney. So the body fluid inside the kidney in the medullary region, that is essential to maintain the hyperosmolarity, and also they should not wash out. They should not wash out to create the to create concentrated urine that kidneys have to maintains the hyperosmolarity. So normal 300 milliosmol per liter. 300 milliosmol per liter. This is the normal high osmolarity of our body fluid. 300 milliosmol per liter. So excess of this, that is called hyperosmolarity, and below that range is called hyperosmolarity. So this is a kidney. This is a nephron. This is a afferent neuron. This is a efferent. So this kidney, this nephron, this is the uh, juxta juxta glomerulus nephron. Juxta. So two type of nephrons are present: cortex nephrons and juxta glomerulus medullary nephrons. This is the juxta. So here, this is a cortex region. All these um, Bowman's capsule, this PCT, this DCT are present in a cortex region. This is the outer medulla, outer medulla, outer medulla. This one is the inner medulla and papillary region. Papillary region. This is the nephron which located inside the kidney. So how the concentration gradient creates. Concentration gradient create first the blood through the afferent neuron, afferent sorry through the afferent arteriole. Through the afferent arteriole, the blood reaches to the Bowman's capsule. Inside the Bowman's capsule, inside the by the glomerulus, blood filters and enter into the filtrate into PCT region, that is a proximal convoluted tube. Proximal convoluted tube, that is called primary solution, primary urine. This primary urine all is equal to our blood plasma. Primary urine is equal to blood plasma except blood cells and some proteins. All those only the in that glomerulus filtration, only large size of molecules will filtered out. All essential, along with water, all essential objects are filtered and sent into the PCT region. PCT region, that is a proximal convoluted tube. Proximal convoluted tube. The fluid the fluid which is present in the PCT region, that is called primary urine. Primary urine. That primary urine contains all essential objects. All essential objects. Those are the here reabsorption takes place. Reabsorption nearly 60 to 65% of that so filtrate, 60 to 65% of filtrate will reabsorb here. Those are the water, bicarbonates, chloride ions, sodium and potassium. These all are reabsorbed and sent into the bloodstream. Sent into the bloodstreams. Remaining these reaches to the descending loop and ascending loop, and also collecting duct. What are the changes will take place in those regions? In those regions, first of all, we should remember that this nephron should start at the time of it start working firstly. When we imagine that this nephron uh, starts working at first time, so what is the changes? First, already we know that the normal osmolarity is 300. 300. Here what should happen? What should will happen here? Along with water, all substances are reabsorbed, means there is no concentration. There is no concentration gradients are created. There is no change of the solid. When they reaches into here, when they reaches into here descending loop, this descending loop is that descending loop is permeable to water only. Permeable to water only. Permeable to water only. This is not permeable to solutes. Those are the uh, ions, chloride ions, sodium ions, potassium ions, bicarbonates, H plus ions are not permitted to reabsorbs. That's why here this loss water, this loss the water only these solutes will be present. So automatically the liquid which reaches here, the solution which reaches here, this ascending loop, this is thick ascending loop, that is in more concentrated, and less amount of water and more amount of ions are present. More amount of those ions are present here. Some specialized cells are present. Some specialized cells are present. Those are the sodium potassium ATPases. Sodium potassium ATPases. Sodium potassium ATPase. What the function of those sodium potassium? These are actively actively washed out the these ions, sodium ions, chloride ions, next potassium ions. These are sent out to the medullary region. So automatically what happens? The constant this interstitial fluid of interstitial interstitial fluid of medullary region will more concentrated than the solution which is present inside the tube. That the concentration gradient is nearly different. Concentration gradient is 200. 200 means here 300. So when it comes to here, that will be in 400. So then here the interstitial fluid of concentration gradient high osmolarities will present 600 600 milliosmol, 600 milliosmol. So 200 difference, 200 here. This one is the 600. This will be in 800. This will be 800. What means the difference is 200 osmolarity. Difference of osmolarity is 800. This 800 means it will be 1000. Okay, 1000 here. 1000 means it will be 1200 milliosmol. 200 milliosmol. This is the high concentration of interstitial fluid of inner medullary region. In the inner medullary region, here when when it comes here, all the solids will last out here and the water last out here. This there is no reabsorption, there is no secretion, and less amount of so less amount of these solutes will reabsorbs. So here the only 6 milli small per liter, 60, and more diluted solution will present in DCT region. So whenever this comes into DCT region and distal convoluted tube, it here is ADH, anti-diuretic, by the influence of ADH, anti-diuretic hormone. Anti-diuretic hormone plays a key role to maintenance to create the concentration, concentrated urine. What is the role of ADH? This ADH will release in the uh, uh, brain, in the area of our brain near the hypothalamus. Whenever here less amount of water will present, less amount of water, more concentrated urine will present than the require. What happens, these cells, specialized cells, will send the signal to the brain, then ADH will release. The ADH will comes into the collecting duct. Collecting duct, what will happen? So the cells which are here at the basolateral side, basolateral side of the cell, there is a receptor is present, that is a serpentine receptor. That serpentine receptor, through that serpentine receptor, this ADH will enter into these cells and create the water pores. So actually, naturally, this collecting duct does not have any water pores will not present. But whenever we need to reabsorb the water, the ADH will enter into the serpentine carrier, then they create the water aquatic pores. Aquatic pores, through the aquatic pores, all will all will water will excess amount of water will reabsorb and send into the uh, uh, in outer medulla and inner medulla. That is the role of anti-diuretic hormone. All these mechanisms is called counter current mechanism. counter current Counter current mechanism. Counter current mechanism means this, the here, so um, the the solution will uh, moves moves opposite direction. This is the descending, this is the ascending. Higher hairpin like structure is present. All these are functionally situated. This is the water permeability, this is the ion permeability. Here there is slightly reabsorption takes place. Main function of here in the near the collecting duct, the ADH, by the influence of ADH, the water will excess amount of water will present, then only the ADH will secrete and reabsorbs the water in the here. All this area from here, from here to here means hairpin and thick ascending loop and distal convoluted tube and collecting duct. It is not permeable to urea. This is not permeable to urea. That is not permeable to urea, that allows that will not allow to comes out the urea. But here when comes the tip of collecting duct, this is highly permeable to urea. Then suddenly the urea will try to escape and reaches into medullary region. But there only will small amount of urea will reabsorbs. It also will create the concentration here in the medullary region. That is called urea recycling. Recycling and ADH. Urea recycling and ADH. These are the three main um, These are the three main to create the concentration gradient in the interstitial fluid. That is the uh, counter current mechanism, urea recycling, and ADH. So this is about the urine concentration mechanism, and also this helps to, that is the counter current mechanism. This creates the concentration. How it maintains? It maintains by the vasa recta. You know, vasa vasa recta. This vasa recta is a specialized blood cells, blood vessels, blood capillaries. Vasa recta is specialized blood capillaries parallel to these loops. So this vasa recta will functions opposite direction means here all the solids will washed out, means will solids will last out last here, and these are the secrets here. So at the same time, this vasa recta blood cells absorbs will absorb these solutions, these sodium, potassium, calcium, and also bicarbonates. All these will reabsorbs, but not here water. When it comes to here opposite direction, this will send out the water, not the solids. Means also here when it moves down, it becomes dilute. When moves up, it becomes concentrated. It maintains the concentration gradients inside the uh, medullary region inside the kidneys. That is the vasa recta. This is the process of urine concentration mechanism. Urine concentration mechanism creates by the counter current mechanism. It also called counter current multiplier. This is the counter current mechanism. So this is the recycling of urea and ADH. All these creates the concentrated interstitial medullary fluid, and uh, inner medulla and outer medulla. This is about the concentration, uh, urine concentration mechanism. So diluted urine and concentrated urine. Diluted urine means when we take lot amount of more amount of water, the body has excess amount of water will present, they filters more, and also diluted more amount diluted urine and more volume of water will present in our urine. So already we had experience. So when we take less amount of water, our urine will becomes thick in color. Thick in why? Because it filters many times, it recycles many times, filters. Then the very less amount of urine will pass, and that will be in very concentration. The volume of water only difference, but here the solids which are the waste materials are equal. Okay, only slightly different. So okay, thank you. In our next class we will discuss one more topic. Okay, thank you.